are from Group B.
Group number 2.
In this video,
We are going to show
about active transport of glucose in small intestine. We perform our result in the form of table that show the content of the test tube, concentrations of glucose, and the percentage of glucose concentration. We determine the concentrations of glucose by using a stack fax machine, and the percentage of glucose concentration can be calculated by using the formula of glucose concentration in the test tube divided by glucose concentration in the control tube times with 100%.
In test tube 1, the tube was filled with glucose renal solution as provided with oxygen. At the end of our experiment, it's observed that the glucose concentration inside the intestine sac is 178.1 mg per 100 ml, while for glucose concentration of the tube is 141 mg per 100 ml. From this, we can see that the glucose concentration of the sac is higher than glucose concentration of the tube. From this, we'll be able to deduce that glucose are transported into the sac from the tube. In normal condition, the mucosal surface of the small intestine of the rat will be on the inside while the serosa will be on the outside. But for this experiment, it will be inverted. Serosa will be on the inside while the mucosal layer on the outside. Hence, the direction of the glucose will be from outside to the inside the sac, which means from the mucosal layer to the serosa and end up inside the sac. Glucose transport in small intestine can be carried out in two mechanisms. The first one is facilitated diffusion, while the second one is secondary active transport. In facilitated diffusion is when carrier protein is used to transport glucose molecules down the concentration gradient, while for the secondary active transport, it is when a transporter protein couples up with the movement of sodium ion and uptake of other molecules. The energy obtained from the movement of sodium ion down the concentration gradient will propel the uptake of other molecules, in our case glucose, against the concentration gradient.
In the test tube 2, which contains glucose renal, 2,4-dinitrophenyl and oxygen, the concentration of glucose in the sac we obtain is 146.5 mg per 100 ml. While the concentration of glucose in the tube we obtain is 153.3 mg per 100 ml. The concentration of glucose in the sac and the tube we obtain are approximately the same. This shows that there are no active transport of glucose molecule from the tube into the sac. Hence, we can conclude that 2,4-dinitrophenyl is a chemical inhibitor which inhibits the occurrence of aerobic respiration, causing no active transport of glucose molecule to take place. So, there are no ATP molecules are produced.
Test tube 3 contains glucose renal with fluorescein and oxygen supply. The concentration of glucose in the sac is 167.2 mg per 100 ml, while glucose concentration in the tube is 147.3 mg per 100 ml. Okay, so the difference from here we can see that the concentration of glucose in the sac is higher than concentration of glucose in the tube. The difference shows that glucose is being transported from tube into the sac. However, the difference between glucose concentration in tube and sac is not as large as the difference in tube 1. This is due to the presence of fluorescein, which has a similar configuration with glucose. Thus, fluorescein acts as a competitive inhibitor. So, glucose needs to compete with fluorescein for the binding site in order to be transported into the sac. That's why glucose can still be transported into the sac, but in a smaller quantity.
In test tube 4, there is only glucose renal solution and small intestine without any supply of oxygen. The concentration in the tube is 166.3 mg per 100 ml, whereas in the sac, is 161.5 milligram per 100 ml. The difference is not significant. Without oxygen, there is no production of ATP in the electron transport chain because oxygen is the final electron acceptor. Without ATP, there is no glucose transportation across the plasma membrane.
Tube 5 is the controlling test tube. This is the controlling test tube that contains of the glucose ringer solutions without the oxygen supply and also the small intestine. Controlling test tube is important for us to know the initial concentrations of the glucose ringer solution that are inserted into the sac as well as in the tube. From our experiments, the glucose concentrations in the controlling test tube is 186.4 milligrams per 100 ml. In conclusion, glucose is transported from the tube into the sac mainly by secondary active transport. And the cell must be kept alive when we tie the knot by supplying oxygen so that respiration can take place and produce ATP energy. From this experiment, we can know that 2,4-dinitrophenol is a chemical inhibitor which inhibits the ATP productions. Meanwhile, furazine is a competitive inhibitor. We have the similar configuration with glucose molecule and they compete for the glucose receptor binding site, thus reducing the rate of glucose transport.